First of all, we'd like to thank Viveka and her team for bringing all of us together for this panel discussion. And the topic is optimizing FR&D to expedite regulatory compliance. If we look at the FR&D activity, we can divide into three parts. One is prototype development, another is getting into analytical evaluations, and the last one is exhibit batches, and then the regulatory filing, etc. If you look at the development part, put together, there are about almost 3,000 companies in India, and about 10,500 manufacturing facilities are there. And these companies could be either API company or API plus dosage form companies, or only dosage form companies. If you look at the development side, in the development, there are different strategies. These companies are having a different strategy. Some companies are driven based on regulatory filings, like some people are having India team, then somebody is having US team, some Japan team, etc., etc. While other companies may be having molecule-based development programs. They will have one molecule based on intellectual property IP scenario. They will start the development accordingly. And in the third case, there are going with the integrated teams where they have people from different disciplines. They form a team and then they do the development. Each way there are merits there. So accordingly, people are going on this. And if we look at the molecule-wise or BCS classification-wise, the challenges are more on BCS class 2 or class 4, and also to the class 3 drugs where inter-subject variability is high, maybe more than 30%. There are the more challenges as compared to class 1 and class 3 drugs. Then we come to the areas, they're like nano formulations or the drug loading or inhalers or mouth dissolving, where the validations are much higher. So there are tools people are using it, and that is what facilitates development scientists. Like for nano formulation, they are using either Dynomill or Nextville, and for analysis, they use Malvern Mastersizer, etc. And if you have to look at the coating on pellets or the tablet part, now there is an inbuilt tool which one can use is Phylon's OCT. And here you can check the thickness. Even if you are doing the loading of pellets or the drug loading, if you are doing it, you can check the thickness using this either online or offline. This is a good tool, basically. And then we come to the bioequivalence studies. In the bioequivalence studies, there are strategies. Now we have Sajeev there. Maybe when we'll have detailed discussion, we will have discussion on the bio study. How do we design the bio studies? Because sometimes before you go to the bio studies, because it is an expensive and time-consuming affair, people go with either gamma scintigraphy studies or detailed dissolution studies, etc. These are some of the tools people are using this, as there are changing regulatory requirements there. And manufacturing-wise, scale up and then take the exhibit batches and then go with either continuous manufacturing or batch-wise manufacturing. Nowadays, continuous manufacturing is the way people are discussing this. But before further discussion on this part, maybe we'll just go with the panel discussion on this so that the discussion is much more useful there. I will go to Ravi Kumar, basically, first part, because the business requests products for multiple markets, but during QTPP, we are having many challenges as we want to develop formulation for multiple markets. Due to various RLDs, in some market one RLD could be there, other market another RLD would be there, and then there are API sources, DMF source, non-DMF source, etc. And FR&D resources are very limited there. Could you please elaborate the challenges we face for such global development program? Request Ravi to throw some light on this.
Thank you, Mr. Arora. At the outset, I would like to thank the Indian Express Group, Vivek and our team for bringing us together to discuss on a topic that's it's very challenging. So I just want to share my thoughts on the question that was posed to me by the moderator. Um, in an ideal world, all of us want to have a global development. We do development of one single product that would meet the requirements of all the markets. But unfortunately, it doesn't happen that way because there are multiple challenges in different countries. So I look at it as a challenge from three perspectives. One is the requirement of the product per se. Two is the regulatory environment and the regulatory requirement that we operate in. And three, the situation, the demand of the current situation. So these are the three aspects that would kind of, dictate the process of global product development. Having said that, how do we optimize the resources of the formulation team? The first and foremost step is to have absolute clarity at the portfolio selection stage. So it's very, very important. Yes, I mean, I do understand that generic business or any other business for that matter is highly dynamic and things change over a period of time. However, clarity with respect to the target on two things. One is the quality target product profile from a technical perspective and the requirement of the markets that this product is intended to cater to. So these two things need to be brought in together as a team. And this comes out of a discussion with multiple stakeholders in the organization, both from technical as well as the business. So once this clarity emerges, the formulation scientist at least has absolute clarity on what needs to be looked at, which markets to be catered to. So that's one. And second thing, Mr. Arora already touched upon, saying that the requirement of the biostudies can be different in different countries, and to make the entries difficult from a generic perspective, the reference product, the innovator, can have two different formulations marketed in two different geographies. So these are certain challenges that are bound to happen. So from that perspective, it's very important that we have clarity on the market. So that's point number one. And the second aspect that we need to look at is from an analytical perspective, different regulatory requirements are there. For example, if you're talking about Brazil, Brazil has a completely different set of requirements for method validations. They need statistical analysis and evaluation, and then we need to have all the multivariate analysis that are supposed to be reported with very, very stringent limits. So that's another cost-consuming and time-consuming effort in case if we are looking at countries like Brazil and China. So this is a very, very important requirement. And this takes, for example, if we are talking about an impurity method development, this can actually be costing very, very high, especially in case of oncology projects, because the impurities are very expensive and the regulatory requirements are very stringent. So that's from analytical perspective. So the third point which I can remember, which I would like to share is, for example, how does it vary from country to country or geography to geography? All of us now, we want to get into the China market being very lucrative. We would like to have a share of the pie there. So we want to enter China, then we'll have to make sure that all the excipients that go into the formulation needs to be compliant with the Chinese pharmacopoeia. And there are certain batch size restrictions or demands that are there. For example, if it's an injectable product, the most preferred thing is that minimum 50-liter batch size should be there. So in case if you're looking at oncology projects, it is not possible always that we have a product with 50-liter batch size. That's a challenge. If it's a tablet, they say that minimum 100,000 tablet requirement is there, even for submission. So these are the challenges that we are looking at if we are looking at a heterogeneous market requirement. And last but not the least, I would like to mention, touch upon the current pandemic situation that we were in. Specifically with respect to the injectable products, where all the resources with respect to manufacturing, the support companies like filter manufacturers, tubing manufacturers, they kind of devoted the complete manufacturing capacity and capability to cater to the vaccine demand. So that had a huge impact on the products that are otherwise normally being manufactured. So the lead times were too high. It went up to even 9 months in certain cases. So how do we factor in those kind of uncertainties in the product development? So can we look at two different set of tubings when we were evaluating, or two different set of filters while the product development is taking place? So these are certain thoughts that we can definitely incorporate in the development process. There's one thing that was certain is that there is going to be a cost implication if we actually adopt to this kind of strategy. And also there is going to be a time implication that we are looking at in terms of catering to different markets. So that's what my thoughts are. And I would leave it to the other panelists to share their thoughts as well. Thank you.
All right. If the time permits, we'll take up this discussion further. But
Sure.
We have lined up a few more questions there. So let's
Sure.
take it up that way.
Sure.
Thank you. There are a number of generic manufacturers having FR&D teams based on regulatory filing, like some may be having India, some may be having China, some may be having Japan, some may be having developing markets, some developed markets, etc. While some companies are working based on the molecules. I have worked in both the areas, both the scenario part. And my experience is a little different there. And some are based on integrated teams. There are merits in each strategy. And what are your views, Dr. Muthu?
Yeah. Good evening, sir.
Good evening.
And thanks to the Express Group for arranging a wonderful virtual meet at this pandemic, to at least to see the faces. Maybe not physically couldn't meet, but at least this is a good platform to meet all the great people around the industry. So coming to the question, what Vinod Ji has asked, and it's a bit not a straight question, and as I think so, this point has been already touched in depth by the earlier panelist, Mr. Ravi Kumar Ji. So from my perspective, if you ask that whether to do a development for the generic products as a molecule based or from integrated process. So if we look at this scenarios, it's not a straight answer, but I can say that there are each and every merits is there in the different way of working, as Vinod Ji has mentioned. Like if we make a development at the molecule base, the merits could be that the particular molecule understanding will be more better to take this product development in a faster pace. And but as rightly Mr. Ravi Kumar Ji has mentioned that there are, other than the formulation development, there are certain analytical requirements for different market. There are some bio requirements for different markets, different. So I would say that it depends on the company. Like assume that the company strategy or the business strategy of the company should be taken into consideration first. Like if the company is not going for a multiple geographies, they are going for only limited 2-3 countries. I think so it should be taken in a different way. And if the company's business strategy is that we need to cover overall geography, it should be taken in a different way. And so coming from the business strategy, if we decide that as a company that we need to go for 2-3 markets, different markets, say US, Europe, and Canada. And so if the clarity has been there during the initial day of development, it will be better for any formulation development scientist to develop the product to meet a stringent requirement. Like if you say US market requirement, we take it because you may have to do multiple bio studies when compared to US, compared to Europe and Canada. And so it is better we develop the product in line with the US requirement, considering other aspects like trade risk for Canada, because the requirements of the Canadian trade risk is that you need to have a similar kind of trade risk. So such physical components has to be addressed during the initial phases. and initiate your development considering second patent aspect as well, and then need to develop a product which will be bioequivalent in the multiple studies like fasting, fed, or steady states, whatever is required for the US market. And this will be the better approach from my perspective. But if you say that some company will be having a global requirement that we need to go for all the market, in that I would say that integrated approach may take a better seat when compared to molecule based. That's all from my side.
Thank you, Muthu. Since I have worked on market-driven basis as well as molecule-based, I felt that molecule-based development possibly gets merit there for two reasons. If you look at the molecule, if you look at the market-wise, people start with India, then they go to Europe in redevelopment, and the Europe team then goes to US development, and then go to another market. So there are multiple developments takes place. Instead of doing that, if you go with the molecule-based development, there are much more merits there. This is what we felt after having experience on both sides.
Okay.
So while your point is valid, saying that yes, for there are both bios are required, fasting and fed is required, while in WHO or other countries only fasting is required, or where the dosage is mentioned that it is to be taken with food, there possibly those details are also required.
Right.
Thank you very much for taking up this topic.
Thank you.
My next question is, we all know that there are challenges with BCS class 2 and class 4 drugs, and also with the BCS class 3 drugs where the inter-subject variability is very high, 30% plus there. Therefore, before initiating the pilot BE studies, what kind of evaluation you do so that the failures are minimized or they are avoided? May I request Dr. Sajeev to throw some light on this?
Yeah, thank you, Vinodi. I think that's a very interesting question. Obviously, yes, the BCS class 3 drugs have unique challenge. So they are soluble, right? So we may believe that, okay, since they are soluble, then they should be easier to formulate, but at times they pose a lot of challenge as far as the bioequivalence goes. So the problem is that though these drugs may be soluble, but their permeability is not that good, or it may be very site specific, right? So the most of the challenge that we face, the challenge in improving the bioequivalence is that they have a very limited window of absorption. Some of them may be getting absorbed in the upper part of the GIT, or some may be in the distal part, right? So they have a limited window. And that's the reason why we face challenge and a lot of variability in bioavailability. So that variability adds up to those challenges, right? So even though the drugs are highly soluble, but still the formulation factors, the excipients, they play a very important role, right? So where the drug is going to get absorbed and whether it will be the same place where the RLD dissolves, that's very important, right? So obviously, the composition itself becomes very important. And let me add here that US FDA has a guidance even on the BCS waiver for class 3 drugs. Though there is a guidance for the class 1 also, and there is a guidance for the class 3 drugs also for bio waiver, but it is actually more tougher in its requirements, because in the BCS class 3 drugs, the bio waiver requires that the product to be having Q1, Q2 equivalence. That means that it has to be qualitatively and quantitatively similar in composition to the RLD, right? So that actually, gives you some idea that having a similar composition as close as possible to the RLD makes, the development a little easier. So that's one point, having composition as close as possible to the reference, if it is possible. Let's say if it is not a patent issue or some other, right? Then obviously the best tool that we have is the dissolution studies before we get into the bioequivalence. So here, as I said, that most of these drugs have very site-specific absorption, so it's very pH dependent. So doing dissolution at the multiple pH values, it becomes very critical, right? So we generally will do, dissolution say at 1.2 or 4.5 pH, 5.5 pH, 6.8 pH, and then observe how it compares with the reference product. So obviously, even the sampling points in the dissolution study, they become critical, right? So most of these are since highly soluble. So we have to keep those sampling points very, very close, in early time points to actually get the idea. So once we have done this type of profiling at multiple pH values, even we can try different RPM, the speed of the paddle and all those things. So that's going to help us. Another point I want to make here is that it's very critical to actually observe the dissolution happening. Many times, scientists they miss out because nowadays companies have become and teams have become too specialized, so that they don't interact with each other. So the dissolution may be happening in another lab by another team, and the formulator may not even know how it's behaving. But the visual observation is actually very critical to see how the RLD tablet or a solid dosage or whatever is dissolving, whether that is forming a heap at the bottom or it is dispersing uniformly in the media, and how the test product is behaving. That becomes very critical, right? Because dissolution can give a lot of information, but you have to use that tool very nicely, wisely as well. So if we can observe how the tablet is disintegrating, how the tablet is dispersing in the media, those observations become very critical. Not just the profile, not just the analytical value, but that observation also becomes very critical. So I always encourage that formulators should go and see when the dissolutions are happening. They should actually stand there and see how the product is dissolving, right? And observe for themselves. That gives a lot of idea. So once all this is done, then obviously we move into the bioequivalence phase, particularly the pilot studies. And most of the times, we face a lot of challenge in, let's say, the FED studies. So again, selecting the meal, selecting the time points, that becomes too critical, because as these are fast dissolving drugs, so there is a possibility of missing out the Cmax value. So having a properly designed pilot study where we can actually capture the Cmax, that becomes important, right? So, but if we see that if the drug is having, let's say, as you said, the ISCV more than 30%, then in those cases, it may be important to look at the design as well. So rather than a simple crossover design, we may have to go for a replicate or a partial replicate design, for such products. So if obviously, if any parameter, either the Cmax or AUC goes beyond 30%, variation, then obviously it becomes a case for replicate study, which makes things much, much easier actually. Though it takes a longer time to do the study and it's a little costlier, but then obviously the success rate is, much higher. But we have to look at the regulatory requirements. In some countries, the replicate designs are not allowed, or you have to really justify why you are using a replicate design. But I think as far as US FDA is concerned, they do accept
Yes,
replicate design. Yeah. So I will say these are the some of the things which do definitely help in designing a BCS class 3 molecule better. And yes, there is a possibility of BCS waiver as well, but the requirements are actually much tighter than the BCS class 1 drug. Yeah. Thank you.
Thank you, Sajeev. You said that the discriminating media is definitely
Yes.
useful for differentiating or checking whether our RLD formulation dissolution as well as test formulation dissolution is similar or not similar. Normally, companies try to have a strategy like formulation wise it should be similar, or the quantitative wise it should be similar. That's normally so that the biofailure is minimal, or the variation F1 or F2 values, whatever you are saying, they are almost, they're closely matching there. What's your thought, Muthu, in this part?
Yeah, I would like to add 2 more points in line with Dr. Sujit, that whenever we make any generic development, specifically a class 2 and class 4 molecule, which is a bit tricky when compared to class 1 and 3 due to its solubility, and in class 4 gets more complex because of the permeability also. But I would say major 2 points apart from what Dr. Sujit has mentioned, that it's about how do you do a reverse engineering of your innovator to get more information about the API characteristics nature, specifically like particle size or polymorphism natures of the innovator specifically, even though you may get some literature information. Reverse engineering, I would say, is one of the major critical point when you start the generic development for any product, but class 2 and 4 becomes little more important part. So first is reverse engineering, and second, coming to that, this point has been already touched by Dr. Sujit, that understanding the site of absorption of the molecules. And if we understand the site of absorption, it will be easy, because there will be some molecules which will be absorbed throughout the GIT, and some molecules may be having a site-specific absorption. So the product which has a site-specific absorption gets more complex, and it's very important to understand the pH of the particular site, or you may require to add some enzymes and you need to do some dissolutions to understand more discriminatory power of that medium when compared to the innovator as well as your test formulation. And when we are able to do this two things like reverse engineering and understanding the product of innovator, and when we are able to discriminate this product by choosing a correct medium, maybe you may have to do extensive dissolution, because it is not a straight away answer. We may have to do an extensive dissolution with respect to the different pH, different RPM, different apparatus, maybe different volume, and then find out a better medium and do a first pilot. Whatever extensive analysis we do, it all gives a preliminary confidence to move ahead for a first pilot. And from first pilot, you can get a lot of information, and then you can go ahead for a IVC to strike in the second deal in case if there is any failure in the first pilot.
Thank you, Muthu. My next question goes to Sajeev. The question is, clinical development program is time consuming, and it is quite challenging as well, and it's quite expensive as well. What challenges you face while planning biostudies? Because the R&D may be different in some countries, and the regulatory requirements are also different. There are formulations either could be lookalike, or they could be scale up, scale down formulation. What strategy you work out when such scenarios are there? What is your experience in this regard? Can you throw some light on this?
Sure. Once again, good evening, everybody, and thank you, Express Pharma Group, for this opportunity to be part of this program here. Now, I agree with you. One size don't fit all. Like, and if it could have, if you could make it fit, there is nothing like that, because then we can derive synergy in terms of cost, effort, as well as in terms of even having the same formulation, same product for multiple geographies. So that is a big advantage if we can work out a strategy around that. But then in my experience, we have a lot of challenges in achieving that for the simple reason that in certain geographies, the strength mix is different. So the B waiver for lower strength, that throws up that challenge. And then you also have the reference product performances varying in different geographies, same manufacturer, same product brand per se, but then the performance wise, they do differ. And then of course, there's a difference in stability requirement from regulatory perspective, but then there is a difference in the packaging configuration in different geographies. So that adds on to the complexities what speakers before me have already alluded to. So from a B study optimization perspective, from increasing decreasing our efforts to sort of get through with the product with minimum B iteration, as Dr. Muthu as well as Dr. Sujit alluded to, like you have to have a very solid strategy for RLD or reference product characterization. And when we talk about reference product characterization, it is just not understanding the qualitative or the quantitative composition of the product, but then most of the time, it is very difficult to identify the particle size of the API in the reference product. But then there are advancements now where you can actually do it through solid phase microscopy as well as hot stage microscopy. You can actually determine. So that gives us a useful input. And then we talk mostly about bulk dissolution strategies. So bulk dissolution essentially means doing it at a 900 ml media volume in type 1, type 2 apparatus. But then people very rarely talk about doing powder dissolutions or doing intrinsic dissolution. So those are another tools which actually help you sort of get very useful insight in terms of what is the performance of RLD. Of course, people talked about multimedia and discriminatory nature of the media. I would go one step forward to have a sort of an optimal BE strategy, and which is where you hit success early on. You probably have to think about the biorelevance of the media also, because GI, if you are talking about the oral product per se, we do have the GI tract throwing us different type of challenges in each of its segments. So if the pH is variable, then the fluid volume, the luminal volume, the fluid volume decreases progressively from esophagus end to the colonic side, or the solid content increases from the upper end to the distal end. You have different gastric transit times. You have to deal with the hydrodynamic pressure that the formulation would face. So from that perspective, relying just on a type 1 or a type 2 apparatus is the old way of doing it. Now people try to do some of these dynamic dissolution strategies like type 3, use of type 3 apparatus more frequently, then also using type 4 apparatus, because there the powder dissolution gives you a very good understanding, because with a closed loop or an open loop system, you can actually manipulate different GI conditions can be simulated much more quickly to understand whether the test formulation is similar or closer to the reference formulation, or is at least expected to be closer in terms of its in vitro performance. If it is closer in terms of its in vitro performance, so there is a very good probability that it would be closer in vivo as well. But then there are certain hidden confounding variables in all these exercises, and those are like when we do a pilot bio, we normally do it at an R&D scale, right? So when an R&D scale typically is, let's say, 500 tablet batches to maybe maximum 4,000, 5,000 batch scale. And what Dr. Ravi talked about with Onco products and all, which are so expensive. So you probably do it at a much very small scale development. So controlling those process variables is very important, and understanding that much ahead of the pilot bio is important, because once you do a pilot bio, get a good result, and when you scale up the process and there are these process variables are introducing certain performance related differences, which normally is not visible in a QC medium, a QC dissolution medium, but then it will only be visible if you have a biorelevant medium to evaluate. So that is another factor that we need to consider when we are looking at BE studies for multiple geographies and for the same product, whether it is a molecule-based integrated approach or a market-driven approach. This knowledge sharing aspect is also important in case the R&D is aligned market-wise, not a molecule or a product, a common product for all things. So then it is important that cross-functional participation from multiple market, the development happening in multiple market, and sharing of that information is very critical. Another, quickly, I would also like to touch upon one very salient aspect that sometimes helps you to accelerate your development program. And that is, if you like, so say for example, you know the molecule is challenging. Like say for example, people talked about BCS class 2 or class 4 molecule being more challenging than maybe a class 1 or a class 3 molecule in terms of its B, the type of B challenges that it throws up. But then can we do some additional dissolution which are not conventional or even biorelevant or discriminatory dissolution, but say, for example, evaluating precipitation kinetics. So to understand whether the challenge is whether we can anticipate certain challenges in the in vivo condition when the molecule will move from one segment of the GI tract to the next segment, and where the pH condition might change or where the ionic concentration might change. So these are, and some of these molecules are, excuse me, some of these molecules are very sensitive to these changes, and then sometimes a very closely resembling product with a very good in vitro similarity with the reference product fails in vivo because we really do not have in-depth understanding about the molecule's behavior under some of these conditions. So these are not classical dissolution methods, but then these are a very useful tool to build a database around the molecule in terms of its anticipated in vivo performance. So these are some of the aspects that most formulators are considering. I mean, gone are the days when it was only a blind trial and error method, but in most companies and most organizations, we do have integrated cross-functional input coming in for almost every development, and there all these aspects are taken into account. But then there are blind spots. Sometimes we overlook certain aspects, and then we end up doing those many extra B studies, which you could have easily avoided if some of this data was proactively generated. And sometimes what happens is that sometimes, you know, my personal perspective on that is, if the SBA of a product, the summary basis approval of a product, is very lean, doesn't give you much too many information, and if the PIL is also very brief on some of these aspects, then you tend to be more cautious right from the beginning, and then you try to generate those additional data, and which many a times prove to be very useful. So these are some of my thoughts in terms of how exactly we, what type of considerations we look at from the product development or from the pre-formulation study or from the dissolution optimization options that we exercise when we try to develop a product. Just one additional point is on the steady state pharmacokinetics that some of the markets require. So when you talk about steady state pharmacokinetics, so matching the Cmax is easy there, but then if you do not match the accumulation kinetics of the test versus reference, so invariably you get surprises when you do steady state at the pivotal stage. Normally during development, you do pilot bios, which are either food effect studies or fasting studies, but you don't do steady state pilot studies anytime, right? So those steady state challenges come up wherein you are not able to meet the accumulation constant there. The accumulation factor comparison is not comparable, or even the Siemens are not matching with the reference product, and that happens because we only in a single dose, when you are comparing, you get the advantage that you monitor the product till the tail part of the pharmacokinetic, I mean the single dose profile. But in case of steady state, you truncate it over the tau period, which could be a 12-hour or a 24-hour. So then if you have a salient difference in your terminal pharmacokinetics from the test versus reference, you end up getting some surprises there. So that is, Mr. Arora, some of the aspects that I thought I should be sharing with the group.
Another thing what can be done is possibly use of gamma scintigraphy studies. Maybe if you have 2, 3, or 4 prototype ready for bio studies, you could consider doing gamma studies, gamma scintigraphy study, and select 1 or 2 for going to bio studies. This is one of the tools we have used it, and we found it, sometimes we have found it useful also.
No, very appropriate for locally acting, for drugs which act locally in the GI tract. Say, for example, mesalamine type of products or other products which work locally in the GI tract and which are in modified release format. Your suggestion is actually a very potent tool to understand where my product is lying in the GI tract during that period, and whether it is reaching the right pH environment to give that, deliver that payload and then work accordingly. So it's very important. Yeah, I agree with you that
we have used this in sustained release product or enteric-coated product, and we found it quite useful.
Right.
Yeah.
All right, we go to the next question here. Another interesting area is differentiated products. When we talk about differentiated product, means where unmet medical needs are like side effects are there. You want to cut down the side effects, or no food effect, or dosage frequency to be reduced, or dose reduction, etc. To be done. What challenges you face in such products from prototype to clinical development and filing? May I request Muthu to please throw some light on this?
Yeah. And so this is one of the big opportunity area, I would say,
Yes.
like differentiated product. And of course, we have challenges, but I would see as a very big opportunity for generic market at this moment. And as you rightly said, Vinodji, how do you increase the or reduce the dose and reduce the side effect? And so there are a lot of formulation technologies which is already available. Like basically, we all know since the drug, whatever you give, is not being properly absorbed from the GIT, the dose has been higher to achieve the maximum potency what is required for the efficacy. So there is always one of the better things what we generally do is increase the solubility of the molecule and increase the permeability of the molecule. So both of these things can be did by different formulation technologies, like by increasing the solubility by using solid dispersion, hot melt extrusions. And if you want to increase the permeability, you can use a lot of permeation enhancers in the formulations. So this will be not very straight away formulation like what we do for a generics. Just we compare the formulation and do a bioequivalent. So this will be a little bit time consuming, challenging from formulation perspective. However, we have a lot of proven technologies which has been used by many of the industries worldwide. And what I feel is that personally, first we need to, when we decide a molecule, and we always select a molecule based on the requirement, when I say based on the unmet clinical needs, rightly what you highlighted. When we choose a molecule, we need to understand the molecule because they are all proven molecules. So we need to understand the pharmacokinetics and pharmacodynamics of the molecule. And our aim is only to alter the solubility and permeability so that we can increase the bioavailability and reduce the dose, and same way you can reduce the side effects. And here we may have to do initial prototype development, what we do for our generic formulation, and then we go for a dissolution studies. Maybe we are not looking for a similarity, we are looking for a higher solubility. Even we can do multiple permeability studies. In vitro permeability studies can be done to check whether our molecule permeability has been increased through the formulations. And then, of course, we need to do a pilot bio studies to check the impact of the formulation, what we have did, whether we have got the desired effect. And this is what we can continue this development, but definitely it will take a time-consuming process.
All right. May I request Sukhi to put some thoughts on this just for 1 minute?
Okay. Yeah, that's tough because this is actually a very complex area.
That's correct. That's correct.
Yeah, but since Dr. Muthu has already touched upon things, so as he said, that PK and PD, they're very important. So actually, before going on with such a development, it's very important to understand what are the PD requirements. What is the target percent? Are we trying to reduce the side effects? Are we trying to reduce the frequency of administration? We are trying to reduce the dose or whatever. Then it has to be linked with some PK properties. Okay, let's say if we are trying to reduce side effects, then we need to understand whether the side effects of the drug are because of, let's say, the Cmax, because there is a peak which is there, the peak concentration, or it is related to the AUC itself, right? So we can design let's say, controlled release formulation in a way where we chop off the Cmax just to reduce the side effects, right? If that helps. So it's very important to have the PK parameters defined. So in terms of, let's say, what is our ideal Cmax? What is the AUC we are targeting? or what is the fluctuation, or let's say in certain products, it could be the C min value. We are trying to target a certain C min value. And then in many drugs, it is time above the minimum effective concentration. Let's say in antibiotics and all, the T over MIC becomes very critical, right? Or in certain other drugs also, like going by my personal experience, we were designing a sort of a control these products for a anti-Parkinsonism drug. And the time above the certain effective concentration was very important, right? So we need to target all these parameters, that what is the ideal range that we want to achieve. And then you design your PK studies accordingly, and then see where where the product is standing. So we need to first fix those parameters. That what is, we can even do simulation studies nowadays. The softwares are available which can do simulations and all. Right. So then go for the actual PK studies, obviously starting with the single dose type of study, fasting or fed, and then the food effect studies. Basically, for such drugs, you need to perform food effect studies to see whether there's any dose dumping which is happening or not. And then, as Sajeev was mentioning about the steady state studies, then you, in between, based on a single dose, again, you can do the simulations for a steady state study. Right? So, as I said, it's very important to actually fix your PK parameters. What is your target? And then, design the PK studies, and then obviously move into the clinical phase.
Thank you.
Yeah.
We have several examples, basically, like fenofibrate is a brilliant example. From 300 milligram to 200 milligram to supraviability 160 to 120 milligram, then clindamycin to Zindaclin formulation, then Flomax to Flomaxtra, which is there are a number of examples there in this particular thing. How about you, Ravi? Would you like to add something here?
Yes, doctor. I think from a slightly different perspective. While Dr. Muthu and Dr. Sajeev, everyone spoke about reducing the side effect or reducing the dose and things like that. I was thinking that differentiated product also could be a delivery system that can improve the patient convenience or patient compliance.
Yes.
So that is one area which I thought that it's good at one's value in terms of differentiated product development. For example, there are many oncology products which have very high doses. For example, 250 milligram, 300 milligram tablets, and then 4 such tablets are to be taken at once, for as a dosage regimen. So already, the patients are suffering, and then we, ask them to pop 4 tablets at once is definitely going to be a challenge. So as a formulator, can we look at something that that can kind of reduce the number of tablets and then increase the patient compliance? It's not actually reducing the dose, but increasing the compliance. Or for that matter, if there is an injection, a 100 milligram injection to be given 3 times reconstitution, it's going to be a challenge for the medical professional who are going to administer the drug, right? 3 different vials, 3 different reconstitutions, injections is going to be a challenge. So can we think of something that is going to help these medical professionals, the paramedicals who are going to administer this dose to the patient? So thereby, they don't do any mistake, and thereby dosage compliance increases. So this is another area which I feel will add immense value. And this can also be a very niche area in terms of a differentiated product category. That's my thought about it.
Adding to what you said, there are basically big challenge on dosing schizophrenic patients, where you want to give the product to them, you don't know whether they have taken it or they have thrown it out, etc. Like risperidone, olanzapine, or aripiprazole, etc. The recent product which was launched, aripiprazole with IEM, that event marker, the ingestible event marker, which was there. That's a way possibly we will see more products coming out in future, which will help people know whether the patient has taken the medication.
Absolutely.
Thank you very much. And I will go to the last question which I have. as generic manufacturers would like to get approval ASAP from generic for their products as per the regulatory strategy. We know some company file with the US FDA and some company file with WHO and other regulatory agencies. Post the issue is on the collaborative CRP basically. Like post WHO approval, one can go with the CRP procedures where you can get approval for the multiple countries in 90 days. While when they file with US FDA, they don't get the approval because there the CRP procedure does not get okay with them. There's another pilot one which is going on with US FDA, which is CRP Lite, which is still not approved. It is still ongoing there. My question is, what type of regulatory strategy we put in so that whatever work we has done in FR&D, we get benefit out of that. May I request Ravi to throw some light on this, and then we'll request Sukhjit and Muthu on this.
Sure. So my thoughts are as follows. While we are talking more about harmonization, the more silos are being created. Now the recent Brexit, that's actually kind of created one more dimension to the entire development activity. Yes, there are scope, but unfortunately, the scope is very limited in the sense that what we call SRA, stringent regulatory authority countries. Once we have approval from these countries, WHO does provide the companies to leverage on that specific bias study or that specific dossier to be used for other countries. But essentially these countries, first of all, the number of countries are limited, I think about 40-45 countries, which is as per the list of the WHO. So only those countries have a kind of collaboration with the WHO, which in turn accepts the recommendations of the WHO based on the original dossier that gets submitted to SRA countries. So that's still in a nascent stage. This was kind of seen in certain situations like PEPFAR program, Mr. Arora, that we went through. So there is, especially with respect to antiretroviral products, there's a slightly wider acceptability in terms of this strategy. Otherwise, I think as a community, we are still to get there. We have unfortunately not been very successful even in harmonizing pharmacopoeia in different geographies. So yes, in small pockets, there are possibilities that we can leverage one dossier that was submitted in one of the SRA countries to the respective regions, but it's going to take time.
Ravi, you well mentioned about this antiretroviral part.
Right.
The challenge is WHO takes much longer time, while the CRP procedure is applicable there. But when you look at the US FDA part, that the PEPFAR program, the approval time is much faster as compared to WHO. Now, question is, if US FDA gives approval there, you can't take advantage of CRP there because that particular program is still in pilot stage. Right. In case if the CRP Lite gets okay from WHO, then possibly people can get much more advantage as compared to what it is there today. Because otherwise, WHO approval is taking much longer time there.
Absolutely.
Muthu, what is your views on the same? The regulatory strategy.
I think so. I do not have any difference of opinion what Mr. Ravi has said.
All right. Sukhjit, your views, please.
Yeah, so
we have very little time left now.
Okay, yeah. So I will agree. So after all this experience of the year, finally we have concluded that there is nothing called a global dossier, and there is no global development as such, right? So unfortunately, still different regions require different. There are some differences, obviously, like we have already discussed. RLDs may be different, packaging may be different, stability requirements are different. So. Yeah, so I believe that even these CRP filing procedure, they have a very limited use as far as now as of now, and I think we need to still wait for a long time to have a harmonized type of things which we can use for a sort of a global filing or a common development. I believe we are far away from that. So As of now, I think we have to develop region by region or country by country. That's the current situation.
But what we are thinking is, if we have a product, if we have a formulation, and if where it can go to maximum, if it can go to maximum number of countries, that's what we are looking at this moment. We understand there cannot be one product which can go to all markets. It could be due to R&D, it could be due to API. Some may require DMF source, some may not require DMF source. Somewhere the biostrategy could be different. Some places it could be only fasting bio which is required. Some places is it fasting and fed, both are required. In some places it is if there are scale up, scale down formulation, you need to do bio on both the strengths. In some places you get waiver there. Those things are there. In some places they will encourage the local manufacturer. If you are taking the batch to a particular location, then those could be encouraged there. Those strategies are definitely there. But definitely one need to maximize FR&D resources to comply to regulatory requirement. See to it that we do the maximum number of filings there with that one particular development program.
Yes.
This is what it is. Now, the first point which was raised was which Ravi had mentioned, saying that if the business will take up that particular part. So in the business request product for multiple markets, but during QTP, we see many challenges as we want to develop formulation for multiple markets, which is due to RLD or API source and FRD resources are limited. Could you please elaborate challenges we face? And Ravi had given the challenges this part. So now I can take up this particular part where Sukhdeep and Muthu mentioned that we may like to say something here. Yes, please.
Yes,
go
ahead. I mean, that's a very interesting question, in fact. So, as I said, as of now, we believe that. So you mentioned about molecule-based teams or R&Ds, or we have, let's say, market-wise, right? So some companies follow a strategy that they'll have a separate development group for US, or separate for Europe, and a separate for other countries like the emerging markets and all that. But there are many it obviously depends on a lot of other factors, their own convenience, and then sometimes managing a very large team itself becomes a challenge in itself. But then maybe it actually stemmed from the fact that when we started off in the generic journey, at that time the regulatory requirements were very, very different. Let's say when you talk about the regulated market or the semi-regulated or emerging markets. But now, as my understanding, that gap is reducing.
Yes.
So today, whether you talk about development for US market or even Russia market or any other market, the regulatory requirements are now, the standards are, every country is raising their standards, and that gap is reducing. So if you ask me personally, I will go for the molecule-based development. That may not actually reduce the overall development time. It's not about time, but then yes, the experience that the team gains, somewhere there could be some synergy. Let's say in analytical development, there could be a bit of commonality. Same resources, same team can be used. Even if we have the same formulation, yes, as I said, that there is nothing called a global dossier. Timelines wise, we may still end up, doing a lot of multiple things on the same molecule for different regions, but still that learning that one gains, the team that gains. So that is common, right? So if that is spread out into different groups, that, in my opinion, is not the right way to go. So in my opinion, now going forward, the regulatory, the differential that used to be there, that is getting reduced. So I will actually go for the molecule-based, sort of organization of the R&D.
That's correct. What do you say, Muthu, on this?
Yeah. So I would like to add one more aspect to that, like molecule base is there, but however, there are some companies which takes with respect to the dosage form also. There will be for oral formulation development, because same molecule will be available in different formulation. So every formulation has different requirements. Like oral dosage formulation has set, you require certain expertise. You need for parentals, you need as additional expertise, and you may be requiring for other dosage form like transdermal or nasal inhalations, other dosage formulations. So there is one more aspect also can be looked from this perspective. And yes, I agree with what Arora ji you have mentioned, and Sujit, molecule based development will give more understanding about the molecule for the scientist to take this product development in a much better way. However, we need to also understand the technology based, means the dosage form based, as well as the regulatory requirements into this consideration, and it depends on the business strategy as well.
I agree with you, Muthu, on this part. If it's a specialized field which is like injectable, or somebody is working on the parenterals part, and the person who is doing the oral dosage form, their understanding may be a little different in both sides. Oral person who is working on the oral dosage form could be different. The person with the parenteral or the topical part would be having some different thinking or different gap altogether. Yes, I agree with you on this part. But if you do the molecule-based development, that is the way where the number of exhibit batches which are being taken by multiple teams could be reduced. And the cost of development could be significantly reduced with that, if you go with molecule-based development part. This is what our thinking was also, and we did observe on this when we were working on the panels.
But I have one additional point there. See, when we talk about molecule-based multiple geography simultaneous development, one very critical aspect is IP alignment. Like, so there has to be a sort of at the initial phase itself, the scope of the project have to be properly defined for all the geographies put together, whatever is of interest. What happens is most of the time those requirements are added on like a train bogie, like a length of the train being increased, like one at a time, every couple of months, a new addition coming in. So that is where the whole thing becomes challenging. So if you can define a holistic scope of the project with IP alignment in terms of the salt or the morph or whatever that is required, because these days IP extensions that come in different geographies, while the filing date might be same for the same patent, but then the patent extension paradigm that sets in different geographies actually shift the launch dates in some cases by several years actually. So from that perspective, the scope of the project has to be defined, keeping in mind the IP scenario, keeping in mind the packaging requirement, keeping in mind the zonal stability requirement. And then of course, for the formulator, you have those challenges of reference product performance being different and all other things. So that is something which have to be experimented and taken care of. But then a definition of scope is very important for such type of development.
Yes, Sajeev, I agree with you. But when you are writing QTPP, that time the IP landscape has to be seen. When you are writing QTPP, you cannot write QTPP unless you have IP landscape, which will give you, these are the markets you can go, these are the markets you cannot go, these are the markets you can only go in these particular years. That will give you the clarity right in the beginning. Then only you can, irrespective of if somebody says you have to go, we want to go into this market, if IP situation is not in your favor, then you will not go there. You need to tell that to the management, look, this is what the challenge is. We may not be able to do this part. So that issue is to be addressed in the beginning, like what you said that, and that almost everybody does that. This is where those who are getting into the developed markets, everybody, whether it is Mankind or whether it is MSN or any company, basically for that matter, everybody does that.
So another point, Ms. Arora, I just wanted to mention is, just to build on Dr. Sujit's point, he was mentioning that the formulation scientist doesn't even know how the erosion pattern of a product is. He doesn't get to see the dissolution performance. So while we all grow, we tend to get into silos. So the trick probably is to break these silos and make sure the knowledge on the product. I mean, we kind of agree that molecule-based development, the global development is going to optimize the formulation resources. While we do that, we also need to break these silos and make sure that the person who is personally developing the product and the method, they need to travel along with the product till commercialization. Unless otherwise that happens, the knowledge transfer is not going to be 100%. I mean, while we say that there should not be biases in the entire process, so we have a separate team that is responsible for doing scale ups or whatever, but the company or the departments will have to be aligned in such a way that these two individuals who have spent a considerable amount of time with the molecule while development, that knowledge needs to be carried throughout and then transferred, and they will have to be till commercialization. That's my last thought on this.
Yeah, and I agree with Mr. Ravi Kumarji, because still I remember in my Ranbaxy days, Vinodji used to say that go and see the dissolution behavior. He always insisted that the person who are doing formulation has to understand the property or the behavior of the product in dissolution. And also, as you rightly mentioned, the person has to completely own the product from the starting of development till commercialization to make the product or the generic product development a successful. Well said, Raviji. Thank you.
Yeah. Ravi, you mentioned, yes, we know some places what people do in the product development, they hand over to process development, and then process development hands over to the production part. At times there are gaps there. Yes, I agree with you on this part. But as you go on, you learn certain things there. You find that the knowledge which was there with the product development guy, the FR&D guys, is not fully transferred to the next part, and the next person who is doing this, the total knowledge the knowledge transfer doesn't take place fully there. So the gaps remains there. If the person who is doing from development to till the commercial part, his ownership is there because it is his baby, and then the tech transfer which is done, it gets what would say that complete transfer of knowledge. His ownership will always be there. Yes, this is my product.
Absolutely.
I agree with you on this part, but certain companies they are not doing, but some they are doing it.
Yeah, but then there is a different perspective also to this. What happens is, while ownership is required, and we anyway, many of the organizations that I know of and I work for actually do the same thing. But then one critical challenge there is at the time of commercialization, when the product is entirely the product manufacturing or technical operations baby, you suddenly see there are certain problems coming up. They are not able to sort of get the same results which probably at the time of exhibit or scale up when R&D team was involved was able to do. And my thought on that is probably because when there you have this change of hands, you have that perfect buyer-seller relationship and questioning of the validity of certain assumptions. So progressively that goes on, get refined, and then product becomes better when that change of hands happen. But then of course, as you mentioned, if the ownership is not there, timeline does suffer because it is nobody's baby. So from timeline perspective and from the agility perspective, a common thread is very critical. But then from a long-term commercialization perspective, because commercialization invariably for all advanced market happens 3 to 4 years from the time of filing, and by the time it is difficult to ensure that everybody who worked in the project is available, and if that is not the case, then it is only the technical operation which is actually responsible for commercializing or the launch of the product. And that is where you start discovering that, oh, if this should have been done or that should have been done. All right, so maybe a sort of an hybrid mode of complete ownership and the change of hand has to be worked out, so that documentation of the information around the product is perfectly done as it moves from the development bench to the scale up to the exhibit to the commercialization stage actually. So that is, I think a more advanced version would be combining these two in a fashion which suits each organization whichever way it can.
Yeah. This molecule-based development plus the integration approach will be the right way to go for.
Yeah.
I think with this, we'll close our discussion of optimizing FR&D to expedite regulatory compliance. We will like to thank Lakshmi, who basically brought all of us together, and she was tirelessly coordinating with all of us to see to it this panel discussion is successful and we all are there together in the same platform.